emotion, and executive function. So finally, we're going to talk about learning and memory. Whenever I talk about memory to a class, however, I always struggle about how to put into words how important memory is in our lives. It just plays a role that is so important in being a human being. We're the only species that has, for example, an ethics of memory. We actually associate an ethics with being able to remember things. Then I read something that was such a beautiful description of memory and so poignant. And I'd really like to share this with you. I've read this many, many times, but it's never failed to just remind me of how important memory is in our lives before we talk about the neurobiology. This is a quote taken from Elie Wiesel, his memoirs, All Rivers Run to the Sea. This is early in his life before he started writing about his memories. He had been a teenager in a concentration camp, and he writes, "Of course I could write my memories of the camp, which I bore within me like a poison. Though I never spoke to anyone about this, it weighed upon me. I thought about it with apprehension day and night. The duty to testify, to offer depositions for history, to serve memory. What would man be without his capacity to remember? Memory is a passion no less powerful or pervasive than love. What does it mean to remember? It is to live in more than one world, to prevent the past from fading, and to call upon the future to illuminate it. It is to revive fragments of existence, to rescue lost beings, to cast harsh light on faces and events, to drive back the sands that cover the surface of things, to combat oblivion, and to reject death. All this I knew, and because I knew it, I told myself I should write. But I had to be patient. Someday, in years to come, I would celebrate memory, but not yet." Just a beautiful description of how important memory is for human life. Here, we're going to focus on the neurobiology. But as I do that and I talk about changes at synapses and everything, think about the role that memory plays in your own life, in your own positive experience of life. In our chapter or our lecture on development, we stress that axons in different systems made connections to other areas of the brain. So we learned that neurons were generated, and then those neurons took up their place and they extended axons. Well, this made it sound as though this was a predetermined sort of thing that happened. And in some ways, it's true that our brain does have some hard wiring, so to speak. So the hard wiring refers to an orchestration predominantly of long axon pathways that make different structures be connected to one another as they should be. So we do have hard wiring in our brain, although we'll come back to just how hardwired these connections are. But if we had to give an example, you remember your eye out here, that the retinal ganglion cells project to a specific nucleus in the thalamus, the lateral geniculate, and then those neurons project to area 17. The neurons in the eye don't project all over. They project to a specific nucleus, and that specific nucleus connects to another. So in some sense, this is a genetically pre-programmed sort of projections that are hardwired or genetically orchestrated. But just as some systems are hardwired, it's equally clear that in humans, much of what happens for us that's important for our behavior and our response to the world is learned. So we could say that it's dependent on experience. So what is going on there? Well, we could have guessed, if we'd known what the limbic system did, we could have guessed that it's going to involve some changes in the limbic system. The limbic system is a system that engages us with the world. So it's going to be the system that's most easily modified by the experience that we have in the world. So the question is, what is it that experience modifies? What is it that's being modified? In what ways are our brains soft-wired? Meaning that they're malleable to changes, that experience influences something that's going on in the brain. Well, we know that we don't generate a lot of new neurons, so it means that something has to be going on in those all-important connections between areas. So that's what we're going to focus on here. We know a great deal about learning and memory, and neurobiologists often use that term learning and memory like it's one word. And the reason we do that is because obviously learning requires some kind of memory component, or you wouldn't have said that you learned something. But most of the research is directed specifically at memory because, in fact, you can have some types of memory without learning. So I'm going to use the term memory, but it means sort of, you'll see, it's easy to follow, that it involves learning and memory, and this is the system that's responsible for that. So what we normally do when we look at memory and learning and memory, memory can be divided into a number of different categories, and these have different functional and temporal features. So first of all, functionally, memory is generally grossly divided into two broad categories. The first is what we call explicit or declarative memory. So explicit memory involves the memory for words, for example. That's called semantic memory, and also the memory of events. So this is called episodic memory. So this is just one type of memory that's involved. There's another type of memory called implicit or non-declarative memory, and this involves a lot about nonverbal memory skills, like motor skills. So for example, skills of being able to brush your teeth. That it's just something that once the pattern gets set into play, you don't even remember when you learned it or how you learned it. So it's called implicit memory. Temporally, memory is commonly divided into three categories. But I want to tell you it's very difficult sometimes to really define the parameters very carefully. But we just do this so that we can study events. There's something called a working memory. And working memory is like an immediate memory. So if you tell me a phone number that I need to know, and I need to remember it for 30 seconds or so I can make the phone call, then this is a very short type of memory that doesn't involve any learning. If you ask me 3 days later, or probably 3 minutes later, what the number is, I can't tell you. So this is called working memory. There's a short-term memory. And short-term memory is necessary for learning to take place. So short-term memory is sort of a memory, and again, the temporal parameters are difficult to specify, but meaning short-term that they don't necessarily last a lifetime. But many short-term memories become long-term memories. And this is the area that we, as yet, don't really know a lot about. But the last type of memory in a temporal sense is long-term memory. So long-term memories can in fact last a lifetime, but I want to emphasize something here. They may last a lifetime, but it doesn't mean that they're not modified by experiences that come after they were made. So memory is a dynamic thing, and so it can undergo change with experience. So in terms of brain structures, what do we think is going on in memory? Well, there are a lot of areas that are involved. These are associated, obviously, in the limbic system. This is the system that's involved in this. Higher order areas of prefrontal cortex, hippocampus, the extrapyramidal motor system, we'll see what that's involved in. That's outside of the limbic system, but we'll see it's involved with a very specific type of memory. Amygdala, structure we haven't talked a lot about, but one that's involved in fear, learned fear, and processing of emotions. And we all know that positive emotions help us remember things and learn things. And negative emotions, if they're too strong, can prevent learning from happening. So it really shouldn't be a surprise that a structure that's involved in emotional processing is involved in this circuit in the limbic system, and also areas of the prefrontal cortex. So let's just talk about a few of these and how they play a role in memory. The prefrontal cortex and this is predominantly areas in this area of the prefrontal cortex, appear to be involved in working memory. So these are the areas that appear to be activated when we have a very short time that we want to have the memory involve some kind of action. So again, it's like remembering a phone number long enough to be able to dial it on a phone. And so this is short-term working memory, and this is predominantly brought about by areas in the prefrontal cortex. We don't know a lot about it. We're learning about it, but we know the main area that's involved. The next area, of course, which I've been saying off and on throughout this course, is the hippocampus, our little seahorse. It's that old three-layered cortex that's involved in learning and memory. And it's going to play a critical role. And it should make sense that an old cortical area would be involved in something like short-term memory, because this must be one of the first cortical systems that developed to help an animal be adaptive. If an animal can't learn from experience, then it can't change its behavior under some other circumstance. And so it makes sense that this old cortical area plays a role. Now in humans, there appears to be a lateralization or specialization of function, and I find this just absolutely fascinating. It turns out that the left hippocampus, so the left hippocampus is primarily involved with memories that involve language. And we note that the left hemisphere seems to be dominant for language. Well, the left hippocampus seems to be really good at remembering words. And the right hippocampus is better at spatial memory. So our ability to find our way through the maze of life. So the spatial memory is more a right hippocampal function. Other studies have shown, and from clinical cases where sadly individuals have had only one hippocampus be involved in some disorder. We've also learned that the left hippocampus, remember I said it was also involved in episodic memory, memory of events that occurred throughout your life? Well, it turns out that the left hippocampus appears to be critically involved in the formation of what is referred to as an autobiography. So what the brain does is construct a story from bits and pieces of events that have happened to you in your life, and it creates a story that internally we see as the story of our lives, that define who we are. What's interesting about that is if you actually check a number of things that have happened to individuals, they're not necessarily accurate, but they're very consistent with how the person views themselves. And so what you have is a creative process that's involved in the creation of your own story of who you are. And we know that there are diseases, for example, Alzheimer's disease, that remove the autobiography from the person. And so we're learning a great deal about that. Now this extrapyramidal motor system, the extrapyramidal motor system, remember extrapyramidal, it's involved in habitual movement? Well, it makes sense. It's involved in that non-declarative type of motor behavior. Like you learn how to brush your teeth and then you don't really have to think about it anymore. So it's a type of learning, and I hope that it makes sense to you that the extrapyramidal motor system would be called into play here because you have these habitual movements that are involved in motor learning. Again, this is amygdala. We've already talked about this. Emotional learning and processing. So it plays a role in our ability to learn and remember because it plays a role in emotions. Association areas of the neocortex. Remember all these areas that were really association areas, so they were either multimodal or they were higher order association areas? Well, we think that long-term memory involves some kind of change actually in protein synthesis in neurons in widespread areas of the cortex. And it's one of the reasons why actually long-term memory, short of massive brain damage, people don't lose their long-term memories generally. Now we'll see in Alzheimer's disease they do, but I'll tell you why at that time when we talk about that lecture. But in general, you don't lose long-term memories, whereas the hippocampus is primarily involved in short-term memory. And the hippocampus is a small area, and if it's damaged, you see very dramatic changes in the individual. So what have neuroscientists learned about what's going on in the brain when we learn something or we remember something? Well, if it's the connections that are involved, then it means that the changes have to occur at synapses. And we call these changes synaptic plasticity, which implies that the synapse is not a static structure, it's a dynamic structure. And synaptic plasticity is a dynamic property of synapses that allow them to be modified and underlie learning and memory. One of the other things we've discovered, which is just fascinating, is that many of the processes that are involved in learning and memory in humans are conserved throughout the animal kingdom. And so this lowly creature, the sea slug, has many things operating that allow him to learn. And the basic mechanisms that allow learning in this animal are the same mechanisms that take place in our brains. And we've sort of modified them or built on these simpler mechanisms, but this is something that's conserved across animal species. Let's think about some ways that synapses can be modified. So let's look at our synapse. Now, we've already talked about some of these, so I'm just going to briefly review this, but I want you to be amazed at how many different ways that synapses can be modified, how they're really dynamic. So, for example, we mentioned that the postsynaptic receptor could be influenced by a nonspecific release of neurotransmitter that might occur out here. Anything, in fact, out here that changes the responsiveness of these receptors will change the membrane potential, perhaps, any number of mechanisms that we talked about. And so if this membrane potential is changed somehow, that means when the normal action potential comes down here, it will be altered. What are some other ways? Well, we've learned a lot about this. Remember that postsynaptic potential, what is generated in the postsynaptic dendrite, can be either increased or decreased. Remember, it's graded and decremental. So the graded part means it's dependent on the amount of neurotransmitter release. So you could have neurotransmitter release go up or down. This is an example of what happens if you facilitate neurotransmitter release. So here's our little rat, and he has an electrode in his brain. We stimulate this synapse, and we get a particular postsynaptic response, and it's that big. Well, it turns out if certain kinds of stimulation are given in between here, and then you go away, and you come back, and you stimulate this presynaptic guy just like you did up here, turns out that the neuron, the synapse, remembers, and now it facilitates the release of neurotransmitter, and your postsynaptic potential is enlarged. That means if you synapsed up here, and now your postsynaptic response is greater, you have more of an effect at the axon hillock than you did before. So your synapses are more efficacious. And so this is one of the main ways, in fact, and this is most highly studied in the hippocampus. So we believe that it's a major way in the hippocampus that learning takes place. And there are a lot of ways of modifying this, but this is the basic mechanism which is involved. Now, another way, let's think about another way we could modify synapses. And all of this is going on in our brain right now in this class. So let's think about another way. Well, let's go back and look at these little things called spines. We didn't focus much on spines. They're these little tiny guys here, these little protuberances of the presynaptic or postsynaptic dendritic membrane. So this is where input comes into a cell, these little dendrites. And the spines are little protuberances of the dendritic surface. So it's going to increase the dendritic surface. That's a main function. But those little spines also isolate synapses. Now this turns out to be very, very important because it turns out spines are dynamic structures, and spines can actually change their shape very rapidly. So there are three kinds of main shapes to spines in the nervous system. One is long, and this is normally the kind of spine that occurs earlier in development. And then in adults, you see primarily stubby spines that are shaped like this, or spines that are called mushroom spines. It turns out that the presence of a significant neck here decreases the effectiveness of that synapse. And it turns out that they can go from one shape to another very rapidly. And so a synapse onto this kind of spine is more efficacious than a synapse onto this spine. And under certain conditions we are still learning about, these spines can rapidly change shape. So when we look at a picture of a synapse like this, here's our main dendritic shaft. Here's the presynaptic component with the vesicles in it. This is the postsynaptic spine. When we take a picture like this, it makes the spine look like a static structure. But I want you to imagine that this little guy right here, which is a stubby spine, can change shape under the influence of experience. And all of these things, changing how efficacious a synapse is, whatever is going on, spine shapes, everything, are these dynamic processes that are going on every minute in your brain, every second in your brain. So those little guys are dynamic. Wow. Are you getting the idea that there's a whole lot of ways you can change things without generating new synapses or generating new neurons? Well, but it turns out, in fact, that we can, in fact, generate new synapses. So in addition to all these changes that take place at a synapse which is already formed, we can also form new synapses. So let's have a look at that. New synapses are formed throughout life, it turns out. And this is something modern neuroscience has uncovered. So here we have our generic neuron. This is where synapses are made onto a dendritic tree, and then an action potential is fired here at the axon hillock and down here. Well, under the influence of what happens to this cell, these end terminals, in fact, are capable of making new synapses. Now, what's interesting about this is there's two different ways. Here I have the lateral geniculate, and you'll remember that input from retinal ganglion cells comes in and could synapse on another long axon cell, which goes out to area 17. But I also talked to you about little interneurons, and it turns out that we think this is where most of new synapses get made, although long axon pathways can form new synapses as well. There's two different ways to do this. This would be where information from the eye, for example, occurs in any pathway, actually makes its synapse on a tiny little neuron whose axon synapses in the nucleus, and then it synapses on a long axon cell that goes out to 17. So this little guy can form new synapses as well. So you could have a long axon pathway form new synapses, or you could have little interneurons form new synapses. So there are multiple ways of even making new synapses in any pathway, whether it's long axon or it's a short axon system. So let's look at a couple of cases in which new synapses can be made. Well, interestingly enough, new synapses can be made after injury. And it's probably one of the reasons why minor brain damage doesn't really change us a lot, which is a good thing. So this may be a way to help us if we have some kind of minor damage to the brain. This is one of the ways we study new synapse formation in the laboratory. So let me explain this diagram. This is normal input to some structure. So you have an axon that forms a synapse here onto a little tiny stubby spine. You have a second axonal input here to another synapse. And so you have two different synapses. Now let's imagine that these axons become damaged. So these axons are damaged. They're going to die and be taken away. They're going to be removed from the system. Well, it turns out this guy who's never been injured, who's perfectly normal, when this postsynaptic area is now devoid of this input, that axon is capable of forming a sprout and forming a synapse onto that site. So new synapses are actually formed. And again, this is one of the main ways that we study synaptic plasticity and study new synapse formation in the laboratory. We can make this happen in the lab. Another way that you have new synapses form is as a result of experience. And these were some just beautiful experiments that were done from beginning in about the late 60s through the present time in neuroscience. It turns out that under certain circumstances, when animals learn new things, you can actually go in and count the number of synapses in their brain. You can count the number of synapses fairly adequately, and you can show that you have action potentials normally. You subject some animals to a learning experience and other animals not to a learning experience. And it turns out that axons can form these growth cones or sprouts and actually induce new synapse formation. So in this case, there hasn't been any kind of damage or anything. There's actually been some kind of experience intercalated between time A and time B. The most incredible demonstrations of this was made by a scientist named Bill Greeno many years ago, in which animals in laboratories are generally kept in small individual cages. And they don't have any social interaction, and they live their lives like a rat lives their life in a very small cage. What he did was just elegant. He took some rats and he made what he called an enriched environment. And he had little toys in sort of a play area. There were a number of animals, so they were socially interacting with each other. Had little wheels where the animals could exercise in these little wheels. And the animals were they were handled by individuals in the lab. Well, it turned out when they examined the brains of these animals, those animals that had been raised in enriched environments had significantly more synapses in a number of structures than animals that were raised in single cages. And this has been confirmed over and over again, that experience modifies the number of synapses in areas like the hippocampus, so that when we learn things, we make new synapses. So we can alter synapses in many ways, but we can also make new synapses. So I just think that is incredible. Now, changes in human beings. What have we discovered in human beings? Well, we believe all the same fundamental things go on in humans. We can change how efficacious a synapse is by changing the amount of neurotransmitter that's released. We can change how responsive a receptor is. We already know that because that's part of what cocaine does. The receptors for dopamine are altered by taking cocaine. So we know that even in the human system that very similar mechanisms are involved. Well, we know something else. We know that new synapses can be made. And we know that even in these long axon pathways it takes place. So, for example, an individual who learns to play the violin, when you look at how their somatosensory cortex is organized, and remember, part of what somatosensory input is about proprioceptive input from the limbs and from structures of the body. When you look at the map of their body in somatosensory cortex, they have an even greater representation of each individual finger of their hand than people who do not play the violin. And this has been shown in pianists and other individuals as well. And so actual experience of life, of learning something, can change what the map even looks like of how the body is represented in the brain. And we're learning that this happens throughout the brain. And lastly, some absolutely fabulous experiments have been done. There's an observation that individuals who have been born blind from birth appear to have like better hearing or better feeling or something, and neuroscientists didn't know what to make of that and everything. Well, it turns out that you can show in animals, and we believe the same thing happens in humans, we can show this functionally, that those pathways that would normally be involved in the visual system, so from retina to geniculate to area 17, we've been able to show that in individuals who are blind from birth, auditory areas develop here in 17 that would have been visual had they not been deprived of visual input. And so what they have are greater numbers of areas that process information in the auditory system and the somatosensory system, so that indeed they may be more sensitive to sound, or the way they're processing sound and processing touch is very different than in an individual who has had sensory input through the visual system. So the brain is clearly very plastic, and it's plastic throughout our lives. It doesn't matter how old you are, you can still learn by one or more of these mechanisms. Thank you.